Good morning. My name is Raul Freitas. I speak from the Brazilian Synchrotron light source. So it's a pleasure to be here. Great responsibility. Very special audience. So I'm here to talk about the recent developments that we have in the Brazilian Synchrotron. More exactly the Sirius light source, which is our new accelerator there. So this is the face of this accelerator. So this is what you see here is the complex that this lab is in. So we have actually 4 national labs here, and the synchrotron is in one of the national labs there. So last time I came here, I was actually speaking about results in this old ring here that we shut down just before we entered the pandemic. So most of the results that I will show, of course, they come from this older machine here, but also I will show you where we are in this new guy here. So for those that doesn't know what's a synchrotron, basically we have this large donut, right? And we have a storage ring that we accelerate electrons, and when the electrons make the curve, we have radiation coming out from these ports. So what we do here is we take one port to extract infrared, and we point this radiation to end stations. In our case, we call this end station IMBUIA, which is the name of a Brazilian tree. And of course, we have a natural acronym here for that. But in this IMBUIA station, we're going to have 2 experimental rooms, one dedicated to nanospectroscopy, IMBUIA nano station, and the other one for classical IR microscopy. So just for you to know, we, right after I came here last time, we started the design and the fabrication of this project, of this beamline. So we took here almost 2 years to, let's say, finish fabrication and purchasing of the components. And then on 2021, we dedicated ourselves to the installation. And since then, we are in the commissioning phase, what we call scientific commissioning phase. So what is that? So before that, just to have an idea on what kind of instrumentation we have there, synchrotrons, they are open user facilities, so we try to have a comprehensive hardware to actually attend this community, which is very diverse and multidisciplinary. So we have there, combined with the synchrotron, we have a full set of lasers, and the idea here is just to combine point spectroscopy with imaging in different spectral ranges. So thanks, Rainer, for the great introduction. So now people everything about is known, right? So I cannot spend that much time here. But now coming back to the beamline, so a few weeks ago, we just managed to extract radiation from that port. So what you see here, it's of course a infrared beam that has also a twist of visible light. It's very nice because we can guide the light to the instrument using this visible part. And this is a big beam compared to lasers, of course, but it's a very nicely shaped beam for synchrotrons. So it's a 1-inch beam that has, this is as we expected in the project, so it means that there is no much deformation of the beam along the vacuum path. We have a little bit of a mustache here that we don't know if it's coming from internal reflection in the source, but the power at low current is more or less what we expected, and we expect to increase this along the next year as we go to the maximum current of this ring. So after we extracted and that's one important point, is that this machine is a new generation of synchrotron machines that are, for the experts here, we know that's very hard to extract low energy radiation from these machines. They're pretty dense, and the magnetic lattice is pretty dense, so it's basically the first greenfield fourth generation machine that has an infrared beamline. So this is pretty nice for the community. So then that's me inside the hutch, and we are now trying to take this beam to the tip, as seems to be quite easy, right? But we actually are on the way to that. So this is an image from a few days ago. So we are now in a shutdown in the ring, so just for maintenance. But we expect to come with the spectra very soon. So I'm sorry I don't have a spectra from this new machine, but this is in the schedule. So okay, so now talking about a few results from the community and also from internal research. So we have, as I will show you, very multidisciplinary results from the community. So this first one is a research dedicated to perovskites. We know that perovskites are pretty important materials for next generation solar cells. So here, basically they were studying using SNOM, more specifically nano-FTIR, to study early stages of degradation of these perovskite films. So first of all, we could see that as we have a broadband beam, we usually do these broadband images, which brings us a kind of broadband reflectivity response of the material. And we could see already some disconnection between topography and this IR signal. And then we investigated the spectral response of those individual grains, and we could see that the band related to the CN stretch in this formamidine molecule is actually changing with this, following this contrast. So as you can see here, this is a film that was naturally exposed to the environment, and we could see these bright spots here. We took spectra on those and also outside, and we could see that the signal here in this CN stretching mode is very different. And we also induced some degradation, so we exposed it to very intense light, white light in these films, and we could see that this degradation is actually changing a lot here. So the conclusion here is that this early stages of degradation happens sort of grain to grain, so it's something that helps people to develop this new material. So this is another example. So here, these collaborators, they were studying new generation batteries, more exactly metalware systems. We know that these systems are key for, let's say, large-scale energy storage, as they are let's say, they hold the highest energy density for any electrochemical device. So here what they did is they combined micro-FTIR and nano-FTIR to study those systems in an open-air cell. And first, they have here an inoperando experiment where they monitored the discharge of the system with the FTIR, so they could see that actually the spectra doesn't change that much with the time, which is a good sign that chemically there is no much change in the cell. But when you go to the nanoscale, although you have, let's say, the same species, the same spectral species there, you have a variation of the intensity relation between those peaks. So maybe we have some heterogeneity at the nanoscale that you don't see in the microscale. So this is a nice work that featured the cover of this magazine. So we were pretty happy to help in this work. This is now another subject, more in biological applications. So people are trying to understand the origins of some degenerative disease related to modification of the secondary structure of proteins. More exactly, here they are studying Alzheimer, which is related to aggregation of beta amyloid to these proteins. And as we have here classically in infrared, we can use this amide band to monitor proteins, and the shoulders of this amide band actually bring some information about the secondary structure of proteins. So this very small shoulder here is the place that we want to actually look closer to see if any change happens when you go from a wild type of neuron to a genetically modified neuron with Alzheimer. So here we combined imaging using lasers. That's a collaboration also with NISPEC. Adrian was there. And we could see that when we isolated these neurons, we can see some different response in these two frequencies here related to special places in the neuron. We still don't have a clear correspondence to this presence in different parts of the neuron, but we could see that at least the wild and the, let's say, the transgenic neuron are different in the spectral response. So now talking about internal research, basically the research that we do in our lab. So it's related to my group. So we have basically three fronts that we work there. Novel materials. So basically we are in this 2D nanophotonics business as well. So we study new materials. We also brought some few contributions to light confinement in nanophotonic crystals like HBN. And now, lately, we are trying to understand more things in the fundamental level of this system. So this is a work that we are developing now, just trying to understand, for example, how we, let's say, modify directivity in the launching of these polaritons, just changing, let's say, experimental parameters like illumination angle and size of this antenna. This is basically, we are trying to help people to think about, let's say, device that you don't have a tip actually there to measure these polaritons. The last example here is still in this field of new materials. We are studying new oxides, so photonic oxides, in this case, tin dioxides, nanobelts. So we call these nanobelts because we actually don't do any lithography in those guys. So we just synthesize those, and we isolate those stripes, and we try to see the nano-optical response of those materials. In this case, both in the IR range and also the far IR range. So the very nice thing here is that this material has some surface phonon polaritons and also volume phonon polaritons. And we were trying to see if this sort of sample preparations is any good for example wave guiding of these waves. So we used Synchrotron we went to ALS to do point spectroscopy and also to take some line scans. So as you can see here in this line scan, we just take several spectra across transverse to one of those nanobelts. So you see here the profile of the nanobelt, and we can see this response, very different response for the two bands here. If we zoom in this part here, we can see that actually we have some oscillations here, some fringes that resemble a cavity. So it means that this material is well organized enough to hold these oscillations here, and with the Fabry-Perot cavity model, we could actually understand this as a real cavity. In this case, a transverse cavity. We could not test the waveguide modes just because they are too long, and also let's say the shape of the end of these nanobelts are not so well defined. So it's very hard to have a nice reflectivity there. We also took some hyperspectral imaging just to confirm those oscillations there, still using the synchrotron, and we could see nicely here the different oscillation peaks, different modes here, and also we could see that above the cutoff frequency for the cavity, we don't see any mode inside. So I will not speak that much about this. So Hans is going to speak more details on these developments. He's going to show other materials as well. So check it out very soon, this presentation. So finally, I just want to give a message here that in synchrotrons we have basically these two pillars of, let's say, action. So we take care of a user's program, and we also do internal research and instrumentation. So basically, we take money from the government, in our case, sometimes a little from private sources, and we keep this program. So we justify this money actually giving this service for the community. But of course, we need expertise to give this service or to give an excellent service. For that, this program actually found our internal research, and that's the way we have these complementary, let's say, pillars that we use to operate those synchrotrons, those open user facilities. But the question here is how to make SNOME, which is a rather sophisticated method, more accessible to non-experts. As you can see, we have like many cases, like a very multidisciplinary. We have people sometimes there that had never seen a AFM in life, so we need to actually understand the scientific case and try to take the most of it, and also more democratic. So in Brazil, for example, we have just few instruments, only one like this, and how to actually serve this community. So basically, synchrotrons in general, they give a full package for users. And in our case, we are trying to improve this service, giving, let's say, during the preparation phase of these proposals, we try to give all the support in sample preparation. This has come already in the proposal. Also to manipulate sophisticated samples, for example, and also to plan the experiment. Second is the beam time. That's the more obvious phase of this process. So we give assistance during the experiment. We keep the infrastructure. We give an instrument that works 24/7 for the users. And finally, we have a post-processing phase, which is key in a sense that people, they take the data, but they need to actually transform that data in publishable information. So specifically for non-experts, this is a phase that they need a lot of attention. So this is just a message that we are working in this initiative. So Ferry here is the driving force between this quasar initiative, and there are many other labs involved. So we are trying to actually make this phase a little bit more user-friendly and more automatic. So if you want to know more information, we can talk later, but we have already some developments to read raw files from this instrument, from the SNOME, into this platform, and to handle this data. So this is something that we want to, in the future, talk to partners here to maybe introduce compressed sensing, deep learning, something that's coming very strongly in this field. So this is a platform that's pretty easy to introduce this. So I want to thank, first of all, my group that's helping me a lot. They are great, and a big list of collaborators and partners, especially NISPEC, that's been a partner for many years with us. This is pretty important for us to keep this, this collaboration, funding agencies, and of course, your attention. Thank you.